bringing back to the stage our founder, Stephen K. Skott. Steve.
Thank you. Thank you. Yeah, I hope, is my mic on? Thank you very much. Fran Lucas's presentation was amazing, but there's two footnotes that I must make. Number one, when he talks about glutathione and cancer, that's your body's glutathione. That is not our product. Our product is a supplement. It's not intended to treat, cure, prevent any disease. However, glutathione does everything. But that's your endogenous glutathione, the glutathione that's inside your body. The other thing I want to point out about his that is quite remarkable, I wish we could get the slide up, is he showed those slides that compared the various types of glutathione. And the only thing that matched hydrostat was intravenous glutathione. That's glutathione that they pump right into your bloodstream. Now that costs about $300 per dose. We cost about $2 a dose. Okay. The other thing is both theirs and ours has the same half-life once it gets into the cell, which is about 90 minutes, which means you get about a 6-hour bang for the buck. Problem is, with IV glutathione, you're not going to get an IV every 6 hours at $200 to $300 per IV. So it's a very, very narrow window. With Swish, you can swish whenever you feel your body needs it. Once a day, twice a day, 3 times a day. And if you swish 3 times a day, you would be spending $6. If you used an IV 3 times a day, you'd be spending minimally $900. The other thing is IV has a problem. And the problem is that the IV does not deliver glutathione into the bloodstream. The IV is in the blood long enough for the blood to break it down into the 3 component parts. Cysteine, glycine, and I want to say glucosamine, glutamic acid. And then once that's in the cell has to reassemble that into the glutathione molecule. That takes time. That takes energy. Your cells don't have a lot of time and they don't have extra energy, but that's what an IV does. Swish comes all ready to go. It's in the bloodstream for 1 pass of the circulation. That's about 50 to 60 seconds. So it doesn't break down, and what gets delivered into the blood with Swish is glutathione. Its cofactor that's critical, cysteine. It's other cofactors that are less critical but very important are vitamin C and curcumin. So just because the bars were as long, don't let that fool you, because to keep the bars as long, you would have to do multiple IVs a day. Very few people can afford that, and very few people like the hour it takes to get that IV, plus the traffic and everything else. So that's kind of ridiculous. Regarding other glutathione, I'm going to share some things after our next speaker that they've asked me to share relating to the Eurofin study. So what you're going to hear next and what follows are some of the most important things you're going to hear, because what you're going to hear separates us from all other glutathione activity. It separates us from all other glutathione and its effectiveness, its speed, its efficiency. So you're going to hear that. But what you're going to hear from my dear friend in a minute is the very foundation, the base that our product sits on, that it works at the base that we'd call a mitochondrial function. It works with the mitochondria and genetic expression in a way that there is no proof or no evidence that any other glutathione does that. He made this discovery. I remember getting a call from Dr. Nikolopoulus, and he said, yeah, go ahead and apply. Now, Dr. Nikolopoulus is a very humble man. He doesn't hype things. He doesn't try to make it bigger than it is, but he also certainly won't make it smaller than it is. And he called me after having put some of his clients on our products. And before he even told me the results he saw, he said, "You have no idea what you have. You can't even imagine what you have." And I said, "Well, I've got a pretty good imagination." He says, "No, no, no. You're thinking way too small. This is such a huge breakthrough, and I can prove it." And you can't. And then when he told me and gave me the evidence of what he saw that nobody else in the world had ever seen with glutathione, he's right. We have a breakthrough that's much bigger than my imagination. We started talking. I flew to Greece. I had the joy, and it is a joy, of meeting this man. This man is humble. You know what's cool about humility? Humble people learn more than arrogant people. They listen, they study, they don't think too highly of themselves, and consequently, they become the smartest in the group. So he's one of the most humble scientists I've ever met, but he's also one of the most brilliant. So please welcome my friend and your partner, Dr. Sotiris Nikolopoulus.
Yo, yeah, hey. All right, stop. Collaborate and I should my brains would something to hold them tight, flow like a hawk, daily and nightly. Go, I don't know. Turn off the lights and I flow to the extreme. I rock a mic like a vandal, light up a stage and wax a chump like a candle. Dance.
Thank you very much. Thank you so much to everybody. I'm so excited to be here. I'd like to thank Steve for the very kind words. But as I told to him, I think myself to be lucky and blessed because I had the opportunity to deal with such technology, the hydrostat technology, and such products like NUMI products. So I want to start. I'm not going to repeat actually what other people said, about NUMI. The technology, you had so many testimonials, from other people. So basically, we're going to go to the science, real science. I want to share the data that we had in my lab all these years that we did it. Basically, it's 2 years that we study NUMI. And at the end, you're going to see what's happening. It's really amazing how this technology works and how these glutathione products give to people. So the title of my talk is going to be NUMI Swiss Effects on Gene Expression. So basically, I want everybody, every single one of you, to understand everything I'm going to say and everything I'm going to present. That's my aim from this talk. So I'm going to start with some introduction, very brief introduction on how cells work. So we are some about 35 trillion cells, every human being. And the way the cells, the way they work is we have our DNA that we inherited from our parents, and that's where our genes are, okay? We have somewhere about 21,000 genes that make up all our cells. These genes, the DNA of these genes is giving the so-called mRNA, which is the active genes, and the mRNA is giving the proteins. So right here, where you see the red letters, there is 4 types of it's different things that basically control how the genes work. These are called epigenetics. It's with red letters. There's the DNA methylation. This is a chemical modification on our DNA. There's histone modifications. There's some other modifications on some proteins that are around the DNA. They're the microRNAs and the long non-copying RNAs. These are molecules that basically, they don't encode for proteins, but they basically control what's coming out from this sequence, DNA, mRNA, and proteins. So this is the epigenetics, which basically control life. Talking about epigenetics, because this is exactly where NUMI products, NUMI Swiss is working. Epigenetics is something, as I said, on our DNA, which is controlled up from so many factors. Actually, it's controlled from the way we live, the environment, the nutrition, if we're taking drugs or not, the disease exposure, if we're exposed to diseases, the environmental factors, if we're next to pollutions, if we're eating pesticides, the microbiome, very, very important epigenetic factor that controls how our genes work. If we're having an athletic life, if we're moving or we're standing and not doing anything, that sport activity is very, very important, a very strong epigenetic factor. Smoking, if you smoke, you basically tell your genes, you stimulate your genes to work in a different way than non-smoking. And this is actually bad, most of them. Psychological status, it's very, very important. It's a different thing to think and have a very good psychology instead of having a very bad psychology. Everybody, the feeling when you're feeling bad, you have a bad psychology, and the feeling when you have a good psychology. Social interactions are very, very important. It's a different thing to your DNA's activity if you're isolated or you have social interactions and you have people that you're caring and loving and all that. And of course, your personality. There are positive thinkers and negative thinkers. The positive thinkers have good things happening on your DNA, on their DNA, so the genes have a good activity. The bad thinkers, the negative thinkers, have detrimental effect on their genes. Every single epigenetic factor that I mentioned, there's lots of bibliography right now, and there's too much research going on the field. So the very first thing that we studied when I was introduced to NUMI was something that is called silent inflammation. So silent or chronic inflammation is a pathology that we cannot feel or perceive, okay? So you can have inflammation for years, 5, 10, maybe 20 years without knowing it because it doesn't give you any signal. But the prolonged levels of inflammation and your inflammatory genes, the pro-inflammatory genes that I'm going to show you later, are associated with any pathology you can imagine, from heart disease to diabetes to arthritis, generally with a very short lifespan. So one of the factors that contributes to the so-called aging, that's why we age, is the so-called silent inflammation. So we look to this with the NUMI products. The definition of inflammation, the silent inflammation, is that the activity of the genes that called pro-inflammatory genes are increased during this process. So how we went after that, how we study silent inflammation, what we did. Basically, we took blood cells, we took whole blood, or we isolated lymphocytes, which is the most important blood cells within the blood. It's a subtype that is a blood that is responsible for our defense. And what we did from this cells population is that we isolated the mRNA from these cells. Okay? So taking the mRNA down, we basically have in a very, very small drop all the active genes, the 21,000 genes that we have in our genome. Okay? And then we selected to look very, very specifically at the genes that are associated with inflammation, the so-called pro-inflammatory genes. So we used a technique, maybe you heard of it because it was very common and very popular during the COVID period, okay, the COVID-19 period, because there were molecular tests that were performed at that time. So this technique is called quantitative real-time PCR. So basically, what is this? We measuring copies of the mRNA of these genes. Okay, the pro-inflammatory genes, when something is very, very active, pro-inflammatory gene is going to have so many copies, lots of copies, mRNAs for this gene. When it's very low, it's going to have low copies of this gene. So basically, we measure the copies of these genes responsible for the inflammation. And before doing that, before measuring exactly what was happening with the NUMI products, I have to say that we worked some years to develop a database basically on pro-inflammatory genes and oxidative stress genes, the antioxidant genes I'm going to show you later, to see how many copies the people have from these genes, the mRNA copies. So we made the database. So far, we have more than 2,500 people. And if you see in here, there are 3 groups that these people are distributed: the high group, the moderate group, and the low group. So each dot that you see, each single black dot that you see, is the measurement of the RNA copies of one individual. So there are around, at this picture, around 1,500 people in there. So you see the 3 groups, high, moderate, and low. We consider the moderate group to be the healthy levels of activity for this gene. Whatever is above the line, the black line that you see in here, whatever is above that, this is going to be the people that have the so-called silent inflammation, increased inflammatory activity of this gene. And whatever you see below this line, this is people that have very low activity of this gene. Basically, the gene is pretty inactive. So for the gene that you see on the left, the COX-2 gene, this is cyclooxygenase-2, very, very important gene for inflammation. What you have when you activate this gene, you have in the production of prostaglandins, and prostaglandins are molecules basically that produce pain and produce inflammation. So we selected from our clients that we have studied one, which is the red dot up here. One of the clients had high activity of this pro-inflammatory gene, and we gave him NUMI. And we tested the same way that we tested him at the beginning. We tested his blood a month after giving him the NUMI switch. And what we found was something pretty astonishing, that his levels went from here down here. So that was a dramatic reduction. If we want to call it in how many times, the scale on the left is on a log scale. So this is like from 2 went down to 5.5. So it's 2.5 to the second, because it's logarithmic scale. So it's like maybe 5-6 times less activity. The same, besides COX-2, we're looking another inflammatory pathway, which is called TNFA. So this is tumor necrosis factor alpha. That's another pro-inflammatory gene that basically we see it many, many times in people that have some heart problems or thrombotic episodes, and generally people that have some circulatory problems. So that was also, we saw the same effect. This person had a very high activity, and after a month of NUMI switch, his activity on that gene went to the normal levels, totally normal. That was one case. Of course, you're going to tell me how many cases you've seen. Well, we repeated this. I was so excited at the beginning. We repeated this again and again and again, maybe more than 30 times so far. And we got the same results. This is another case of another client which had two different pro-inflammatory genes. One was the interleukin-6, which is called IL-6 in here on the left, and the interleukin-8. This is for IL-8, interleukin-8. Both of these are cytokines, very, very important pro-inflammatory genes that we basically see them in absolutely so-called chronic inflammation. So they don't give you any sign for that. So in both cases, both IL-6 and IL-8, the people that were in the high levels, so they have the genes overexpressed, they basically downregulated their expression, and they became normal for the activity of these genes. And another client, which I'm showing here, two different cytokines, which are two different pro-inflammatory pathways. One is interleukin-12 on the left and interleukin-1 beta on the right. So the results were interleukin-12 from a high level, it went down to a normal level. And interleukin-1 beta, that's a control basically. It was at the normal levels, nothing happened. It stayed at the same level. So whatever is high is getting normalized with glutathione from Yumi, and whatever is normal, it stays normal, which is excellent. This is a perfect control. Okay, so the next section of the talk is about the oxidative stress. So oxidative stress, by definition, is the accumulation of the free radicals. What are free radicals? Free radicals are byproducts from chemical reactions. These chemical reactions occur in all our cells, all our 35 trillion cells, and they accumulate. Okay, this is an internal and physiologic problem. Okay, but what's going to happen if you have, let's say, exposure to more bad sources, like if you eat processed food, if you had exposure to pesticides, if you have exposure to radiation, if you're smoking, if you're drinking alcohol, all these all causing more oxidative stress. So besides the internal, that's so-called all these free radicals, the internal production of the free radicals, you have more free radicals produced from the exogenous sources. So what's the problem then? The problem is that you're damaging your cells continuously and more efficiently. So, of course, God gave us glutathione, okay? But it turns out that glutathione, the way it's produced from the body, is never enough. So we're short of glutathione, and we always need more glutathione to do the job. I have a presentation here because many people asking me that they don't understand what exactly is oxidative stress. They basically they confused with the stress that we have in our lives. You know, like I'm a very stressful person, and so on. So I give a very, very simple example, to understand what's happening in the cell and what's happening with a tomato. So a tomato, when it ripes, it's getting bad and bad and bad and bad until it becomes totally waste. And what's going to happen after that? We just throw it in the garbage. The same thing is happening as we age. Okay, that's where aging comes in here. With our cells and the free radicals produced from the endogenous or the exogenous sources that I described before. Okay, you have attacking of our cells and our molecules. Okay, our cells, our molecules, the DNA, the RNA, and the proteins from these free radicals. And the damages, if they stay there, are very, very strong. And basically the cell is collapsing. So maybe you heard about the terms of some cells ghosts, the ghosty cells, which are basically cells that lost everything. That's just garbage, but they recycling inside our body. And this is one of the very, very bad things that's happening when we age. So I have also here a schematic to show how important it is to have your antioxidants very, very strong and to have everything in balance. So the whole life, if we want to make a sketch about what is life, is basically a balance. A balance of what? A balance of these called epigenetic negative factors, all the things that I described at the beginning, the poor diet, all these pollutions, the drugs, the chemicals, whatever, and which is the red scale of the scale, the red side. And on the right side, which is the green side, we have all these antioxidants that we normally have in our bodies, which we have it at the highest degree when we are born and we're very young, but they were diminishing. By definition, they diminishing as we age. Glutathione, of course, is on the top, but there are also other antioxidants like catalase and superoxide dismutase. Very, very important also antioxidants that protect us and keep this balance at the right position. So when this balance is going towards the red side, so the weight is more to the red, what's going to happen is for sure a pathology. Okay, that's how important it is to maintain this balance at the horizontal and very balanced way. So, talking about oxidative stress and these antioxidant genes, we went after some clients that we found to have very low activity of these oxidative stress genes. The one on the left is a client that we discovered him to have very low activity of its catalase gene, okay? And we gave him Numi for 14 days and 30 days. So we took his blood 14 days after and 30 days after. And we checked again what's happening to the activity of this gene, this antioxidant gene. And what we found is from a very low level, he went to the perfect levels that we found in our database. Okay, so this happened both 14 days, which is the green, or 30 days, exactly the same measurement after 30 days of using Numi. So we saw the effect 14 days. That's very, very short. We haven't seen this because we studied other supplements and other molecules, I would say, what's happening to these genes. And it's the first time we've seen something working so fast in 2 weeks. The same for catalase, we showed with another antioxidant gene, which is called SO2. SO2 stands for superoxide dismutase. Very, very also important antioxidant gene like glutathione is. So we got a person here. He gave us the blood to see what's happening even the 14 days or 30 days after he got it, the new switch. And as you see, his results became normal. Same thing with catalase. Thank you so much. Thank you. I'm going to finish very, very shortly. You know, I don't want to spend more time. This is another 2 results. This is with interleukin-6 receptor. That's another gene, very, very important also for the activity, antioxidant activity. Very, very low amounts from this person. They went to normal again in 2 weeks and 1 month after switching with Numi. And as in the first example that I said, I showed to you with the pro-inflammatory gene, for this antioxidant gene, which is the GPX1, this is glutathione peroxidase. It's the enzyme basically which handles glutathione when it enters the cell. This client had totally normal levels, okay? It's the red. So you see, nothing happened. It remained the same. It's exactly what we found for the pro-inflammatory genes. We found also for the antioxidant genes. So the pro-inflammatory genes were low, were high, the pro-inflammatory genes, and they went normal. They got reduced in their expression, and the antioxidant genes were low and they went high at the normal levels. So you see, it's like a balance. Glutathione does a balanced job. This is amazing. This is incredible. We haven't seen this multi-action at the same time. Thank you. So my conclusion here is that from this short talk, I mean, I could stand here and talk to you forever, but I don't want to, take your time for that. I conclude that NumiSwiss is a powerful modulator. I would call it modulator because it modulates high to normal, low to normal, of the expression of genes involved both in chronic inflammation and oxidative stress, which is basically everything. Thank you very much. Thank you so much. Thank you. Thank you. Thank you.
Okay. I'm going to repeat.
That's okay. Thank you. Thank you. Thank you. Thank you.
He is so humble that he takes something that is mind-boggling and in saying, wow, nothing else in the world can do this. He says, nothing else in the world can do this. There's the difference between our personalities. When he told me that he was seeing things that nothing else does, you see, if you've got something so powerful that is lowering the expression, the negative high expression of one gene, it's very apt to have collateral damage and lower the expression of genes you don't want lowered. That's normal. We are normalizing underexpressing and overexpressing at the same time. Now, you've never seen that before. As an expert who's seen everything he could see, he's never seen that. That's what he got excited about. And there's reasons that we're doing that, and no other glutathione product in the world, no matter how expensive, no matter how it's packaged, does that or has proven. They won't even try this. And we didn't encourage him to try this. He just saw such changes with himself and with his clients that he wanted to see why it was working the way he did. He had one dentist, right? 72. And she could not hardly even walk down the stairs of her home. And how long was she on it?
How long?
How long was she on it before things happened?
2 months?
2 months. And then all of a sudden, she can walk down the stairs. She's excited. And he starts seeing all this. So he's got to measure it. What is going on? You're not going to get this anywhere else. And so, I mean, it was important enough to me that I flew to Greece. And then I find out we had already had phone conversations, and I was already feeling his spirit. And then we meet face to face, and everything I thought about him was confirmed. You know why humble people have the opportunity to become the smartest in the room? Because they listen before they speak. They read before they speak. They collect before they speak. And they see certain patterns and then they speak. But then all of a sudden, something that would be very disruptive comes along, and there's a whole new pattern that's better. And what you have going on here is I've never seen before. Nobody has this. You don't have any idea how big this breakthrough is. Nobody has ever said that to me. He said it because it was an absolute fact. I had no idea what he was seeing. So we got together, we saw it. He mentioned silent inflammation. Another member of our medical board, his name is, he's a good friend for many years, Doug Harrington. Doug Harrington is a brilliant research scientist from Stanford University. He's brought over 200 therapies, cardiovascular and cancer therapies, to the world. He's had a team of 60 other Stanford scientists working with him. Brilliant man. He's the one that said, everybody else's product is like a skateboard and you've got an F-35. That's Doug Harrington. He saw this and he went crazy. And he says, let me tell you why I'm so crazy about what he's discovering. How many of you have heard of plaque in the arteries causing a heart attack? Raise your hand. Plaque, calcified plaque. Okay, that's all you hear about. That's only 8% of the heart attacks. The 72% is caused by what they call invisible plaque, jelly plaque, soft plaque. It hides in the lining, whereas calcified plaque is actually in the vessel. Soft plaque is in the lining of the vessel. And you don't know it's there, and your doctor doesn't know it's there. But silent inflammation causes it to grow until it's like a pimple inside your vessel. That pimple pops, and you who've never had heart disease are dead on the floor, and the doctor doesn't have an explanation. That is 72% of the heart attacks in America. Okay? Silent inflammation causes a hidden plaque. What he is seeing with glutathione is he is seeing a management of silent inflammation, which is the exact same thing that our scientists from Stanford saw. And he couldn't explain why. He explains why. There are actually people out there that have glutathione products that sell for twice as much as what ours are. And they are on the internet saying, ours doesn't work. You can't get glutathione from a mouthwash. Okay? They're wrong. They are so dead wrong. And we have the proof we have the experiential and anecdotal proof every time we give glutathione away, we start hearing things. But now, between his study and Eurofin's study, we have proof that nobody else has. And so I was asked to just touch on the Eurofin study here. You can stay here with me because there was a slide missing in my presentation. We had so many changes in that presentation going for it, back and forth, that the final one was missing a slide. So who's ever doing the slide, if you've got my Eurofin slides, let's bring them up. That's me. Okay. What I showed you yesterday was this slide. And these are various glutathiones. This glutathione is normal glutathione. That's L-glutathione. This glutathione is called double E glutathione, and it is specially prepared for cellular penetration. And you can see what it did. This was hydrostat. Now, this is in the first 5 minutes of exposure to a cell. 5 minutes. You can see we blow it away. Then, at 10 minutes, this one drops from 56 to 41. This one drops from 75 to 27. They're going down. They're not going to work. This one goes from 110 to 150. No, nobody has what we have. That's what I'm telling you. Do you know this drives me crazy? This is why I got up out of a room, walked, and a scientist was talking to an empty chair. Aaron was in the room, so he was also talking to Aaron. And now these are normal cells. Normal cells aren't our problem. You know why? I should say these are healthy, unstressed cells because they don't need more glutathione. We're giving them the frosting on the cake. What needs our product even more, desperately needs glutathione, are the distress cells. And the older you get, the more distress cells you have. When you're young, when you're a child, you might have asthma and have distress cells in your lung, and everything else is a healthy cell. When you get old, you're getting distress cells everywhere you turn, your muscle cells, your joint cells, your heart cells, your brain cells. But look what happened with distress cells, and this is what we did not show you yesterday. Go to that slide, please. Here we go. Are we going there? Has the slide changed? Oh, okay, here we are. So these are distress cells. These cells in the lab, they reduced them to 35% less glutathione in the cells. They did that chemically. They go below that, the cell would die. So this is normal glutathione in 5 minutes. Let's see what happens with the specially prepared glutathione. Oops, it went up a little bit. What happened with hydrostat? Whoa! 300% more than this, almost 400% more than the specially prepared glutathione. Now watch what happens after 10 minutes. 30 drops to 26. This one is going up a little bit to 37. What happened to hydrostat? Nobody has that. Now, somebody said, well, this isn't an independent lab because NUMMI paid for the test. When you take your car to a car dealership because the engine has stopped working and the tires have gone flat, you don't need, you're going to pay for it to fix it. Okay? Yes, no, this huge organization is not going to do it without being paid. But we could pay them $10 million and it wouldn't change their test results one iota. These are scientists and technicians. They're not saying how good are we. Their simple summation was what happens when hydrostat is exposed to the cell. Now, what this test doesn't show is how do these other things get to the cell. This one never gets to the cell because it's destroyed in the stomach. This one never gets to the cell because it's destroyed in the stomach. Aha. What do we call our product? Swish! Because it goes right into the bloodstream through the mucous membranes in our cheeks and in the vascular, in the cavity underneath our tongue. So we not only see results when they get to the cell, we actually get to the cell. That's what you have. Does it work? Talk to anybody you give the talk to an athlete. We have lots of athletes now. We have doctors that work primarily with athletes. Guess what? We delay the onset of lactic acidosis in a conditioned runner 16.7%. I asked Dr. Osorio here in Spain, I said, is that good? It doesn't sound that good. He says, is that good? Nothing else changes at 1%. This is the biggest game changer I've ever seen. He said, this is why our athletes on your product recover so much more quickly. It's why athletes that won't recover at certain levels at all are recovering now. So we see the experience, but the experience is validated by this, and it's understood by this. We're working at a mitochondrial level in a way that an expert has never seen work before. That's why he says, we got so much more to do. There are things that we can do with our technology that nobody else can do. And we talked the other night that got me all excited because I'm going to be 78 years old. And he said, you know, there's two genes that if we could just turn the switch on, there's a group of genes, you'll get another 20, 30 years. Do you think that's worth working on? Would you like those little genes switched on? We're going to work on it. He's going to work on it. We're going to do it together. Thank you. We have three of our scientists that do a lot of work for us because they've seen the results. Doug Harrington from Stanford, Gordon Crozier, many of you know him. He is the head scientist for the anti-aging medicine. He's been with me from day one, and now we have Sotiris. And this is our future, not our past. Our past makes the future possible. Thank you, Bert. Thank you, God. And thank you guys for giving us the money and the success to keep the work going. But we have a future. And you're not going to see this kind of a future in any other company in the world. I'll give you one more little tip. You know why we see the redox level? Redox, remember, redox is how you keep the gun loaded. Okay? Here comes a bunch of free radicals. I've got a machine gun. Uh-oh, I'm out of bullets. Here's more free radicals. I don't have any bullets. We're the only glutathione product that keeps reloading the gun because we include cysteine. We put cysteine in the cell, free cysteine, L-cysteine. We're putting into the cell at the same time we're putting the glutathione in. So keeps reloading. Okay? That's what we have. That's what nobody else has. So you can look at any glutathione bottle in the market and you're just going to see glutathione. What else do we have? Guess what the power for all that is? It's called hydrostat. We're the only ones that have hydrostat. Thank you. Thank you, my brother. There you go.
Scribe and 124th Street.